So welcome back, everybody. Happy Monday. How was last Friday? Is it okay? Should I do that more frequently? You guys. I wasn't here. No, I'm going to be here most of the time. That was scheduled before the semester started, and I wasn't able to move it. So thank you for your patience. I'm glad all of you, it looked like all of you or most of you completed the assignments that I gave you for doing in class on Friday, even though you weren't here. Today we're going to move on with these topics and talk about, briefly, because most of you have had this in previous classes, but again, I want to make sure that everybody's up to speed on what we know and what we don't know about nucleic acids in their various forms. So this is going to be the first sort of more traditional class that you've seen in this class so far. We're going to talk about some definitions, some concepts, some basics, laying the groundwork. But at the end, we're going to do something totally different. Hopefully everybody's in this talking mood and brought some opinions on the reading assignment that we had for today. The journal, or the magazine article rather, on the Olympian and the lady that have this shared mutation. So hopefully everybody at least glanced at that. When we started class last time, I asked everybody while you're waiting, instead of a quiz like we did today, was tell me what your first thing you think of is when you hear the word or see the word mutant. Pre-association. Close your eyes when I say the word mutant. What's the first thing you think of? An integral aspect of the evolution process. I don't know if that's pre-association. I believe X-Men, Teenage Mutant Ninja Turtles. I think of corn, horrible mutant teosinte. It also doesn't sound to me like free association, but yes. So all of these are good examples of, and of course I can't argue with your opinions, what you think of when you think of the word mutation. Of course, maybe the answers here were tainted a bit by the fact that you're sitting in a genetics class, so maybe everybody was in a genetics frame of mind when they gave these answers. Evolution, evolution, evolution. I'm good with that. Okay, so one of the things that we're going to be talking about a lot in class this term is mutations, mutants. Is anybody in here a mutant? Maybe I should do this as a Socrative because we've got one in the back, I'm one too. Everybody really is genetically unique, so technically we're all mutants. It's kind of like a mutant support group. Hi, my name is Dr. Ross, and I'm a mutant. Step one is acknowledging that I've got, well, I don't know if it's a problem. So to drive this home, I did what I usually do and what I'll advise all of you to do in class when I ask you for an image that represents something or when we do something creative or artistic. The first thing I do is go to Google Images, images.google.com, and I search for, in this case, the word mutant. And like some of you, the first thing that comes up are Teenage Mutant Ninja Turtles, from the Aliens, from the Aliens movie, some Aliens, Mutant Ninja Turtles. I don't know what the rest of these horrible looking things are. Interesting to note that apparently, according to Google, because you know how Google ranks the images it shows you, it's kind of who clicks on which images the most, which ones are most frequently viewed for these search terms. We've got categories like human mutant and real mutant people. Who's searching for actually, I'm probably the one that searches for real mutant people to show you images in class. So these are the sorts of things we're going to be talking about this term. We start with DNA, the molecule, understanding how DNA is structured, what it's for, how it reproduces, how it replicates, how mutations happen, and then towards the end of the term, the latter half of the term, we start talking about how those mutations affect organisms. By the way, before we go any farther, Brandon, can you stand up or raise your hand, please? Brandon Ortega is a graduate student in my lab, and he's going to be our grader for the semester. So you'll see him in here. He was a superstar in genetics back in his day when he was sitting in here amongst you. And so you'll see Brandon a lot, and again, he'll be doing grading for us this semester. So welcome, Brandon. All right, so now it's time to take a look at the results of this quiz that you just took. So the first question, if I could remember what the first question was. chromosome contains 15% A. What percent G does it have? 68% of you got it right. So of all of the questions, this is the one that was done least well on, according to the results. So on average, the class gets a D. Well, B, I guess, in this class. So how do we work through this then? So this is low enough results that it's worth briefly going over. What's the concept that we're talking about here? Chargaff's rule. So if you've got a DNA molecule that has 15% A, how do we work out the percentages of the other three nucleotides, T, C, and G? Okay, so A's and T's are going to be the same. Why? Because it's Chargaff's rule. No. Hydrogen bonding. So everybody hopefully will remember that A's on one strand of DNA base pair using 2 hydrogen bonds to T's, and that G's hydrogen bond with 3 bonds to C's. So if there are 2 DNA molecules here, that would look like that. So there will always be the same number of A's and T's in a molecule of DNA. So how do we do the next step? Right. This has to total 100%. So we've got 30% accounted for already, so we know that G and C have to account for 70% of the nucleotides in this chromosome. So we have to divide that by half to get the number of C's, or the percent of C's and the percent of G's. So in this case, the answer was 35% G was the question. What percent G is in this molecule? Okay. So two-thirds of you got it right the first time. Now we've seen an example of how to apply Chargaff's rule to a type of question I would probably ask on an exam. Everybody did very well on DNA structure in general. The next 3 questions, which direction do you read a DNA strand? Was it a DNA or an RNA molecule? It was DNA. So the only other question that we need to go over then is the hydrogen bonding question. So if we have a molecule that's got a sequence CGAT, how many hydrogen bonds will this DNA sequence make with the opposite strand? So how many hydrogen bonds does the C make? 3. And the G? 3. And the A, 2. And the T, 2. So that should have been 10. Okay. But it's clear, given the results, that everybody remembers your introductory biology, so we don't have to spend much more time on the basics of DNA structure. Are there any questions, though, about DNA structure, polarity, 5', 3' notation? Because like I said, we're here for you to ask me questions for me to answer to improve clarity of these concepts. So anything you read in the textbook that's confusing, you want to see examples of DNA polarity questions, DNA structure, difference between DNA and RNA. It's going to be really boring if nobody comes with questions. If you don't want to ask out loud, you can always tweet me questions at Ross Biology. Then no one knows you're asking a question. It just shows up in my feed, and I'm kind of sly. Oh, that's a good question. Wait for that tweet. No questions? Okay. Do raise your hand when you have one, though. We talked about Chargaff's rule. Now we get to move on to what some of you, only got time to show a couple of images that you submitted when I asked you to send me images of DNA where I should have put photos of DNA. I should have, like I said on Google Classroom, been more specific and ask you specifically for photographs and not drawings, diagrams, etc. Those abound. But if there's one, I'm glad you're listening. If there's one thing that the students in genetics never, very rarely, understand completely by the end of the semester, which is so critical, is in this image in the bottom left. What are you looking at?
Mitosis.
This is mitosis, which involves, what are these hot dog things? They're chromosomes. This is what I figured, and I'm glad, because most of you that posted a photograph, which is okay if you didn't post a photograph, you still get credit for doing the assignment. My bad on not being very clear on the assignment. Photograph of chromosomes. This is kind of what I was expecting. So we've got chromosomes. Pictures of DNA. Right? Somebody was sitting in a microscope, they were looking at a cell, or undergoing mitosis or meiosis. And so we take a picture of DNA. There it is. What's in the upper right-hand corner? Some of you might recognize this. It's kind of a picture of DNA. I don't know. You get credit for doing this, but this was the first time anybody looked at the structure of DNA. Rosalind Franklin, Watson, and Crick. Somebody said something right here?
Oh, I just said Rosalind Franklin.
Yeah, Rosalind Franklin. So, right, the unsung hero of the discovery of the double helical structure of DNA. Take X-ray diffraction patterns, and if you're an X-ray diffraction pattern kind of person, you can look at that and say, oh, of course, DNA has a double helical structure. So, I'll leave it to the experts to convince me of that. But so, yeah, it counts as a picture of DNA. It's not what we see under the microscope, though. This is kind of what I was expecting. Again, looking through a microscope at metaphase, part of the cell cycle. We'll talk about the cell cycle in a few class meetings. So chromosomes at metaphase, part of cell division, one of the stages, right? Prophase, metaphase, anaphase, telophase. This is where the chromosomes are really compacted, so they're easy to see under the microscope. And then they've been colored using some fancy molecular techniques that we'll talk about as we go through the term. I played detective a little bit, and I think I figured out, after we do this, I think I figured out how most of you came upon, the vast majority of you found just one photograph of DNA. I think I know how you did it. But first, let's do this. So give me a second before you get onto Socrative again. I want to end the original quiz. Okay. So we've got a multiple choice question. It's one object that was in that photo that was submitted. How many molecules of DNA are there? Really? No one's going to go for 8?
How do I log back into Socrative?
Oh, yeah, same room number in case you didn't. So 240820.
240820?
240820 is the room number. Always for the whole semester, it's fine. You'll memorize it at some point. So no one's going to go for 8? Okay, but we've got a race going. Very few of you are getting distracted by the distractor choices. So I'm glad to see that you figured out how to take tests. I'll rule them out for you. It's not 0 and it's not 8. Any technical issues going on right now? Raise your hand. Okay. So we're sort of stabilizing. A quarter of you have said 1. A quarter of you have said 4, and 50% of you essentially have said 2. Okay. Turn to your neighbor, and in 2 minutes, convince your neighbor that you got the right answer. Okay, go. Either explain why you think you're right, or your other option for us to move forward is to ask me a question that will help you figure out whether or not you got the right answer.
So he took my option away. You guys are lucky. Oh, you got a question.
I do.
Do the telomeres have anything to do with how you figure that out?
They can. So when you know the number of telomeres, that helps you figure out how many molecules of DNA. So how many telomeres are up there?
4.
So if you were really carefully reading, which you didn't have to be, on the previous slide when we showed the full picture, underneath in the caption it said that the telomeres were labeled in green, those green dots up there. There's 4 telomeres. I feel like there might be a root problem here that's causing confusion, or maybe lack of confusion. Maybe I'm making this too difficult.
You have 2 chromatids connected at the kinetic cord, or with kinetic cord, are they considered 2 molecules or 1?
Critical question. What do we call a molecule here? What's a molecule of DNA? If I ask you to draw me what a molecule of DNA looks like, what would you draw?
Would that be like a phosphate, a sugar, and a nitrogen base?
So a nucleotide would be like the pentose sugar with a nitrogenous base, the phosphate, hydroxyl. Okay. Then you could connect another nitrogenous or another pentose sugar with another base to that. So what's this whole thing? Is that a molecule of DNA? Got 2 nucleotides connected to each other through the sugar phosphate backbone? I'm not just asking for how you want to define a molecule. Then I'll adapt how I proceed the rest of the term to what you all tell me is a molecule of DNA. If you don't like this, how would you change it to make it fit what you envision a molecule of DNA being? So where do the hydrogen bonds occur? This is what we call one strand of DNA. It's got that sugar phosphate backbone, or I forgot to add a phosphate in there. So the sugar phosphate backbone with the nitrogen spaces, the ends, hanging out into space. This is where those hydrogen bonds occur. So if those are hydrogen bonds, what nucleotide is the N for any nucleotide? What nucleotide is this one on the bottom? If it's making 3 hydrogen bonds. It's either a C or a G. The one on the top is either an A or a T. So is one strand of DNA a molecule, or do we have to draw the other strand to make it a molecule? What do you want? Because only after we figure out the definition of a molecule can we figure out the answer to this quiz question. How many molecules of DNA are there? So are we going to call one strand one molecule? You want 2 strands? Okay, so I see some heads nodding. Thank you for that. So let's see. If the phosphate in the upper left-hand corner, which carbon is that attached to? We're talking about nucleotide polarity now. 5 primes. The phosphate's always connected to the 5 prime carbon. So if I was going to draw the opposite strand, you already know, because you told me on the quiz when we started, the 5 prime is going to be at the top or the bottom of the right strand that's not drawn yet. Okay, so 5 prime is going to be down there. The 3 prime end is going to be up there. And so if that N happens to be an A, then there's going to be a T, it's space paired over there. And if that happens to be a G, there's a C. So now you have double-stranded DNA. 2 strands, hydrogen bonded together via the nucleotides, the nitrogenous bases. So is this a molecule of DNA? Okay. What's another way we normally represent a molecule of DNA then? Double-stranded DNA. What does it look like? What did Rosalind Franklin discover? Looks like a double helix. So 2 strands of DNA base paired together. We'll call that one molecule of DNA for class. Everybody comfortable with that? So that's going to be our working definition. 1 molecule, 1 double-stranded piece of DNA. Okay, so 1 molecule of DNA is going to be 1 double helix. So like I said, this is the image that the vast majority of you, not all of you of course, but the vast majority of you found this image when I asked, show me a photograph of DNA. Scanning electron microscope image of DNA. Except it turns out it's not, but we'll get to that in a second. So what do all these red arrows show, do you think? They've blown up a part of this molecule, maybe, of DNA, and it looks kind of like a screw, lots of little thin threads. What do you think this is evidence of? It kind of looks like that. How do the authors interpret that image of DNA? Take a picture of DNA, looks like that. What does it mean? Do you think? Don't read the text at the top, that's cheating. What does it mean? There's a regular pattern along the length of this molecule that they photographed. DNA is supposed to be a double helix. They suggest this is consistent with DNA's helical structure, because nobody's taken such a high-resolution picture of DNA before, and so they're trying to say, yeah, Rosalind Franklin's zany interpretation of that X-ray diffraction pattern was right. DNA is helical. Except, at least one of you submitted this picture. It's the visual abstract from a journal manuscript that was published on this. And interesting, this is not new. This is 2012 stuff. This isn't like groundbreaking today. This was 4 years ago groundbreaking. That molecule that they took a picture of was not a single molecule, a double helix. It was 6 double helices that were all assembled around a 7th double helix in the middle. So it was a bundle of 7 molecules of DNA. And they took a picture of that. Still, reasonable submission for literally a photograph of DNA. So when you do, and I'm assuming this is how most of you found this, I Google image the term DNA photograph. Most of what I get is either drawings or it's this picture in various forms. So good use of Google Images for those of you that did it that way. So that all works for what a photograph of DNA looks like. We've got metaphase chromosomes, and then we've got these long stretched out electron microscopy photographs of DNA. So in the coming days and weeks, we'll talk more about the structure of DNA and what chromosomes are, how they function. The next thing I want to talk about, and the last topic to talk about in class, is this article that I asked everybody to read. Yes, it was long. hopefully you got some interesting things out of it, because these are important to understand. So can somebody give a quick, like, half-minute synopsis of what this paper, what this article said? What was the basic point? You're going to tell your parents about this at dinner tonight, or significant other, or kids. What would you tell them in 20 to 30 seconds?
Well, it was kind of basically these two women up there had related diseases, and it was very rare. And back, I guess the woman on the right, she's the one that discovered it. She kind of researched herself because I believe the doctors hadn't really known yet what it was, and they didn't really rule in that women could get it more susceptibly than men could. And then she kind of researched it herself, dug deeper, and then she kind of found a research lab that would take her blood and stuff. So.
That's right. I thought that was a particularly interesting part of the article. But so can you send your blood to Italy in the mail? It turns out, yeah. When the Italian research team emails you and says, hey, can you collect blood from you and all of your relatives and send it to us? You can apparently do that. So that's one thing that the subject of the story did that was perhaps unique or unexpected. What else did she do that was unique or unexpected? What traits does she have that makes her, even though she is not a biologist, a successful geneticist? I mean, she's, as far as I know, never took a genetics class. Might have. She did all this research on the internet.
She was determined.
She was determined, probably motivated for a personal reason. Sir?
She has a disease, so it was easy for her to know the symptoms and stuff, right?
Right. So she knew her own case better than any doctor ever would because she was living it. Yep.
Thorough. Very thorough through the investigation.
Right. So she did a lot of reading on the internet, though. I mean, that was her tool. She was not at an academic institution. She didn't have the Henry Madden Library necessarily. Right? She didn't have access to all of the scientific journals because, as some of you may have noticed, if you ever tried to get access to some of these journal manuscripts, sometimes you get this page that comes up on the web that says, please pay $32 for 12 hours of access to this. And you think, what? Right, so she probably encountered the same thing. So she was stuck to accessing only data that was free on the internet. So she was invested. She had a reason to be interested. She had the internet. What else? What sort of other characteristics? Interesting things happened in the story. What traits did she show that made her successful in diagnosing her own genetic disease? Yeah.
She diagnosed herself with an extremely rare case of this muscular dystrophy, as well as her dad kind of made the connection about the cardiovascular, and then this track and field athlete.
Right.
Has a similar genetic mutation.
And yet looks very different from the woman on the right. So this makes a critical point. So there are many reasons that I asked you to read this paper and that we're having this discussion right now, not just to kill the last 12 minutes of class. One of the points to take away from this, no, I'm serious, I'm not just trying to kill the last 12 minutes of class. One of the points to take away from this is that these two women share a genetic mutation, yet their phenotypes are totally different, right? We've got lack of muscles and fat on the right. We've got huge musculature and also lack of fat on the left. So they both have this lipodystrophy disease, lipodystrophy, fat breakdown, or absence of fat. Take home point number one. The same mutation or mutations in the same gene can cause very different diseases. Right? They share the fact that they have mutations in the same gene, but those two mutations, they're different mutations, do totally different things to the people that have those mutations. So just because you know they have mutations and that they're in the same gene doesn't really, in itself, tell you anything about what the women look like or what sort of symptoms they have. Now, sir, you also mentioned the fact that this impacts multiple organ systems. So for all of you, what sort of systems besides fat and musculature? You already said this. Well, part of the muscular system. Which organ is particularly susceptible to damage? So there's loss of fat. This woman on the right has very little musculature in her arms and legs. And what else does this disease predispose to? Pardon?
Respiratory failure.
So it was organ failure, heart failure was one of the key things. Is that right? That was what she was worried about with her dad. So how does one mutate? So this is within the family now. The daughter and her father share this mutation, this really rare mutation. It's hereditary, that's why he could pass it to her. But he was at risk for heart disease, they discovered, and she is, but wasn't as severely. So take-home point about genetics, number 2, is a conversation we'll carry on through the semester. The same mutation, one mutation in this family, in one gene, can cause multiple effects that seem to be totally unrelated to each other. I mean, this mutation causes an issue with heart function as well as in other systems as well. So it's not like you get one mutation, you get one disorder. You get one mutation, it can have a suite of effects, what physicians call syndromes, right? Lots of different effects that don't appear to have anything in common, but they are, in this case at least, caused by one mutation. So I'd like everybody to go to this website, please. We're just going to explore briefly. I want to introduce you to OMIM. It's a great resource to use. You can use it on exams if you want to. Of course, when we get to exams, you can use anything on the internet if you want to, but this is a good resource to draw on. omim.org. Online Mendelian Inheritance in Man. This is a human-curated database of every hereditary genetic condition that affects humans. So if there's something published about a genetic disorder, you can learn about it here. Right? Wikipedia is good too. Use Wikipedia. But you can also use OMIM. It's outstanding. So let's look up the gene that is mutated in this woman. It was called lamin, one of the lamins. So its gene symbol, you can search by the name of the disease too if you want. What was it called? I don't remember. Let's look up LMNA. It's the gene symbol for lamin A. You could just type in lamin A. So here are some probably, hopefully, familiar terms. Number 5, familial partial lipodystrophy. We've got muscular dystrophy, right? Some terms that were used quite a bit in this article. So if you click on one of these links, it will give you lots more information about those specific diseases and those specific genetic mutations.
What are the numbers mean?
The 1, 2, 3, 4, 5, 6, or
No, no,
the
no.
longer numbers, the 6. See, you guys should be asking these questions. I'm glad you're here. So each of those 6-digit numbers is just an accession number. So every entry in OMIM gets a unique numerical identifier. There is a difference between the asterisks and the number signs, though, or you might know as hashtags or pound signs, depending on which generation you're from. Right? So the asterisks are for genes. So lamin AC is a gene.
Yeah.
The number signs are for diseases.
The hashtags.
Yeah, or hashtag, yeah, number sign, pound sign, hashtags, whatever. So if we click on the 150330 lamin AC, that brings up this huge record, everything that's known, basically summarized for you about this gene. The only thing I'll point out at this point, you can learn a lot about it here, is its cytogenetic location. So it tells you on which chromosome and where on the chromosome this gene is found. Just one of many pieces of information that would be useful to look up about genes. It's an easy way to get information about this. omim.org. So this is on chromosome 1. That's what circled up there. 1q22, we'll talk about the nomenclature in a while, but the first number, 1, is the human chromosome it's on. So this is a mutation in a gene that's on chromosome 1. Any questions? I just wanted to introduce you to one resource to use when you want to look up more information about genetics as we go through the term.
Can I ask them a question?
You can ask anybody.
Who knows what that means, chromosome 1? Who can tell me? Because I don't know.
How many chromosomes do we have?
I don't
We
know.
humans. 23. Stick around. You're welcome to come back to class after you're done being the guide on the go, because we're going to get into the human cytogenetics and chromosome descriptions in about a month. But to answer the question, chromosome 1 is chromosome 1 because it's the largest chromosome.
Okay.
So in humans, chromosomes are numbered from largest to smallest.
Okay. Okay. I'll just ask you.
That's, okay, so at this point, there's one more conversation to be had. I just want to wrap up to make sure that everybody's comfortable with the goals for today. So we've discussed a little bit of vocabulary. We came to a consensus. I hope you feel like we came to a consensus on the definition of a molecule of DNA being one double helix. You already came with understanding of DNA polarity, nucleotide structure. We talked briefly about Chargaff's rule, and now I want to get to that last question. So we've talked a little bit about genetics and society, how to be a successful geneticist, right? It's useful if you have a personal connection to what you're studying, which many of you probably do in this class. You might not know it yet. It's helpful to have the internet as a resource, which is why I like to teach this class as a discovery class, because we can use the internet during class and during exams and after class to learn interesting things. But the question I'd like to end with today is, what did this article about the lamin mutations, muscular dystrophy, lipodystrophy, what did that tell us about society and ethics? What ethical issues were raised in this paper?
Well, for some, like, when she went to one of the doctors about saying that she thought she had this type of disease, she's like, no, you definitely don't have it, you're female. So, I know, she should have probably looked more into it rather than just like, no, you don't have it.
You would always hope that well, at least I shouldn't say you should. I would always hope that my physician would be open to my assistance. And I actually, personally, honestly have in the past diagnosed myself, not with genetic conditions, but with other things. It took me a long time to get a physician to actually say, yeah, you might be right, let's do this test. So ethical for physicians. If you're a doctor, you've got this huge caseload and you're trying to see patients as quickly as possible, would you rule out, yeah, I know you think you've got that, but it's only ever been reported in men? It's a great ethical question, something to consider, even if you're not interested in being a physician as a career. Other ethical issues? What would you do if somebody contacted you or your agent in this case and said, I think you should get genetically tested to see if we share this mutation? Right? Think, I don't know if I want to talk to this person. I'm trying to get my DNA information. This is a huge ethical issue.
Or what if your brother called you or your sister and said, I just got told that I have this disorder genetically. Mom and dad got tested, but they don't have it. But you should get tested.
Oh, and then we've got then we get into all sorts of
Yep.
related topics.
Sorry
We'll
about that.
we'll get into. So the thing that I found, and I know we're almost up, 1 minute, last point. So what did this athlete, how did she respond when the primary person that this article was about started telling her about how she felt as a kid when kids teased her and said, yeah, they think my mom doesn't feed me and so forth. How did the athlete respond? What did she say? What were her experiences as a kid or as an athlete?
She connected like almost instantly to the stories that she was telling.
She related to the stories. So what sort of things happened to her when she was a kid or when she was a practicing athlete?
She got rumors for like, oh, she takes steroids and
Right.
stuff.
So people thought, oh, this athlete takes steroids. So now she's been diagnosed with a mutation. Does that mean she should be able to compete? Should she be able to compete with other women? Should she be able to compete with men? If all of us have mutations, we're all genetically unique. It's kind of a gray area, right? How do you look at somebody's genetics and say, oh, you should be able to compete with these people, but not with those people because you have this mutation that makes you extra muscular, right? How is this going to be applied to sports? So think about it. For next class, we've got no reading assignment. So come to class, we'll talk about prokaryotic chromosome structure.